Very good afternoon again. So, as I told you that after the class of composting, it is very essential to understand that since we are living in a modern days of agriculture, so farmers are must trying to do the measures that are quite helpful for the environment. See, whenever you have seen the farmers are utilizing the chemical fertilizers, I hope that you understand how the application of urea has create a drastic changes in our environment. You know, whenever these chemicals are being applied or supplied in a crop field, it is okay that they are there to provide the essential nutrients in the form of nitrogen, phosphorus, potassium, calcium, etc. But after you get a good growth of the plant, we never think about the soil conditions, the microbiota of that soil. You know, microbes can't differentiate. They are going to act upon those chemical fertilizers, and that is in return are actually harming the growth of microbes. So dear students, we have to find out an alternative, and I know that in most of your places peoples are using the alternative to those chemical fertilizers. So in this lecture I am going to talk about biofertilizers. So what you are going to learn from this topic, the the things which you have studied from last few weeks of this BTY 164, now you are going to apply it. You have seen the role of microorganisms very well in the development of composting also. You know, biofertilizers is nothing new. It is again a kind of requirement of soil which you are providing with the help of microorganisms. So students, let's move on from to the next here to the next one. So actually, what exactly you can say to them, they are the nutrient inputs of biological origin for plant growth. Definitely you are providing them for the plant growth. So they are the nutrient inputs of biological origin, right? So examples that you have already seen, so many discussion goes on in your lab experiments also. Even you have isolate rhizobium, acetobacter, pseudomonas, cyanobacteria, all these things students. Here you can see in the pictures also the last this particular part where the person is using gloves and he's collecting the azolla, right? This is a cyanobacteria which is being taken from the rice field, right? The water collected from the rice field. So this is being used as a, all these species are being used as a biofertilizer. Okay, so next one. All right. So yes, what are the nutrients you require from them for the plant growth? They are essential nutrients such as we are going to categorize them first, right? As I have discussed many a times, even in lab classes also, that we are categorizing our nutrients into two categories, either macronutrients or micronutrients. Micronutrients are those which are required in very less amount. Macronutrients are those which are required in large amount. Okay. So if you see over here, the categorization can be clearly seen in this slide. Macronutrients: carbon, hydrogen, oxygen, nitrogen, phosphorus. Dear students, I have said this many a time, CHONP. If you remember this code, I think you are never going to forget this. CHONP is actually the macronutrients. It is required in good amount. Carbon is an essential, dear students. You know, we all are built up of on a carbon frame. Our all biomolecules have a essential carbon backbone. Either you consider carbohydrate, either you consider amino acid, proteins, lipids, etc., etc. Micronutrients, for example, it is written iron, manganese, boron, copper, zinc. You know, these are required in very minute amounts, but yes, they are very essential. Why? Because many enzymes of metabolic pathways in plants requires these essential nutrients which are in micro amount. Is it clear? So these inorganic ions are acting as a catalyst of enzyme. Oh, sorry, they are acting as a cofactor of enzyme. So without them, enzyme could not function, and if enzyme could not function, the metabolic pathway could not proceed. Right, students? So let's move on to the next one. Right. So far, if you have seen fertilizer, so what exactly it is? The fertilizer is any material of natural or synthetic origin either you see urea, which is a synthetic one, and natural, as I have just discussed, right. So when it is applied to soil, for the supplying of one or more nutrients, that helps in the growth of plant. This is the simplest way you can define. So if somebody asks you fertilizer, yes, they are of chemical or synthetic or natural origin, where when they are applied to the soil to provide the any nutrients for that which is required for that plant, I mean for growth, right. So if I am talking about the chemical synthesis of nitrogen fertilizer, if you see, yes, this is somewhat maybe new to you, maybe you have studied previously also. If you see in this slide the Haber's process, right. So if you remember Haber, I mean the picture is quite well depicted over here. See, this is the process of industrial nitrogen fixation also, where N2 and H2 leads to the formation of ammonia, and in this process the iron is acting as a catalyst, right. So this is a chemical way of synthesizing some nitrogen fertilizer. This is just a repetition of Haber's process, which I have discussed. Okay, so let's move on. Yes, why one more thing, that why you are using such kind of fertilizer, because it gives good yield obviously, but the problem is it is non eco friendly, it is destroying microbes, and they are expensive also. So we must try to find out the alternative. Fine. So just now I have discussed that if you want to overlook those disadvantages, I mean you can find out an alternative, which are nutrient inputs of biological origin for plant growth. So if you see over here, all of them are in the natural origin of things. I mean they are obviously having chemical constituents in them, but they are of biological origin. Okay, so let's move on to the next one. So biofertilizers, exactly how you want to define biofertilizer, it is the natural fertilizers which are prepared of microbial inoculants such as bacteria, algae, fungi, etc., maybe separately or in combination also. It helps in biological nitrogen fixation or phosphate solubilization. You know that phosphate availability to plants is somewhat difficult because it is not easily solubilized. So microorganisms are required to get it solubilized and let them provide it to the plants. They also helps in the building of soil microflora, because I told you that if microbes act on the chemical fertilizers, they are going to be disturbed or destroyed. So when these biofertilizers interacts with soil microflora, so they help in maintaining the healthiness of the soil. They also include certain organic fertilizers like manures, etc. So definitely it is always being recommended by people who are doing the organic farming. Fine. So you have heard about organic farming, where people are not using chemicals, instead they are using the natural origin components, right. So yes, what are benefits? Definitely they are eco-friendly, they are increasing soil fertility, they are increasing the crop yield, they are of low cost and easy to grow, and farmers can easily grow blue green algae. They are not that creating any issues, and yes, they are resistant to plant pathogens also. Okay. So here I have categorized them into few parts: nitrogen fixers, phosphate solubilizers, mycorrhiza, green manuring. Dear students, nitrogen fixers, we already know that there are various categories of nitrogen fixers which are living in symbiotic or asymbiotic. So if first I will describe nitrogen fixers, you can see over here this first yellow line on the left hand side, it is showing symbiotic and asymbiotic. I have already discussed this many a times, even many other faculties have discussed this, that symbiotic nitrogen fixer is rhizobium, one of the best example, and asymbiotic is acetobacter, azolla, etc. These things already has been discussed in the lab, and you have isolated them also in different media. Fine. Second is phosphate solubilizers, where Pseudomonas, etc., etc. Aspergillus, Pseudomonas is a bacteria, Aspergillus is a fungi. So they are helping in phosphate solubilization and making them available to the plants. Mycorrhiza, as I have discussed previously also, it is the association of fungi and roots of higher plant, where they are providing the phosphate to the plant, while in return plant is providing the nutrients, the shelter also to the fungi. And then green manuring, where you are using most of the things which are of organic origin, right. So in this you are using certain microbes like Sesbania, Rethyrus, Tephrosia, etc. So Symbiotic nitrogen fixer, you know already, rhizobium. This we have discussed in our lab parts also, that you are going to isolate on Yama medium, that is yeast extract mannitol agar. The composition of this medium has already been discussed in the lab part also. So if you see that how rhizobium appears onto the medium plate, you can see that this rhizobium appears as somewhat, you can see over here, white and glassy somewhat. If you see the nodules of the plant, you can see the presence of rhizobium in these nodules. So these are the nodular part that I have described also earlier in the lab part, and in this nodule basically rhizobium resides. It lives in symbiotic associationship. Okay. So, asymbiotic acetobacter you have already described and you have done it in the lab. I mean the isolation of acetobacter on Ashby's medium, where it is appearing a very smooth, appearance, fine. So, they are not living in symbiotic associationship, they are simply living in non-symbiotic, they are free living. It means they are helping plant, but not living in associationship, they are living in free living format, right. So, their role is to fix the atmospheric nitrogen. Here I have shown that in the form of arrow, nitrogen helping by are basically fixed by microbes. In what form? In a form which is taken up by plant, that is ammonia, fine. So, not only ammonia, in fact nitrites and nitrates also. Phosphate solubilizers, which I have discussed also, that the rock phosphates which are present over there in the soil, it is not solubilized easily, it could not make available to the plant, and if phosphate could not be made available to the plants, it means there may be certain difficulties in some metabolic pathways. So, those rock phosphates are being solubilized by microbes to help in the formation of soluble phosphate. Examples: Bacillus, Pseudomonas, Micrococcus, Aspergillus, right. So, they make sure that phosphate solubilizing bacteria can be isolated in Picosskaya media. The Picosskaya media composition is given here in the left hand side, and you have to remember this. And if you see on Picosskaya media, if you see the growth of microbe, the zone around the central part of inoculated microbe, it is clearly showing that this microbe is able to degrade phosphate, fine. So, because it secretes that phosphate degrading enzyme, fine. So, that is why it helps in the solubilization of phosphate, and due to that it is able to provide the phosphate to the plant. Okay. So, I will just move on to the next one. So, what exactly is their role? When I am talking about bioremediation, you have to remember three things: bioremediation, biocontrol, biofertilizers, and at last phytostimulation. I will discuss each one. Bioremediation is actually the cleaning up of the contaminated soil, right. By using these microbes or biofertilizers, you are helping in the cleaning up of the soils. Then biocontrol, with the help of this microbe, you are actually attacking the plant pathogens, which is also a microbe. So, this is the microbe against microbe, that is biocontrol, right. Then biofertilizers, in order to improve the nutrient efficacy in plants, you are using this, which I have just discussed. Now, what is phytostimulation? Phytostimulation is production of indole acetic acid. I mean, indirectly they are helping the growth of the plant. So, indole acetic acid is one such hormone which helps in the growth, and then increasing the rooting, increasing nitrogen uptake, reducing fertilizer uses. These are some phytostimulation. So, students, remember biofertilizer, control, phytostimulation, and bioremediation. All these are being taken care by the microbes, right. So, if you have any doubts, I will be available on Zoom from 2 to 3, no worries. So, how will you actually do that? I mean, the cultivation of those biofertilizers. If you, I hope you have seen autoclave in the lab, that is, these are the instruments required for sterilization. You have used laminar airflow chamber, you have used incubators, you have not used bioreactor, but in bioreactor you are actually enhancing their production, right. You want some very good production in a very shorter duration of time. So, this is large scale cultivation in bioreactor. So, do not forget these instruments. Okay. So, they are being applied for the production of biofertilizers. Biofertilizer from nodule isolation, you have done this. I don't need to repeat it again. This is the, I mean, you have been discussed this last time with your faculty. So, this is the nodule sterilization, and then from nodule you have isolated the uh, rhizobium. If you see on the rhizobium, means I have shown you in the Yama medium few slides back how it appears, right. So, this is the same experiment I have discussed many a times also. I have shown the slides also where rhizobium has been grown on the Yama medium, that is yeast extract mentor agar. This is just a diagrammatic representation of how to start with, right. So, you can see that first of all you have to take the culture broth, you have to grow it at a large scale, then you are going to check it. Now, then you are going to provide packager. I mean, there must be a carrier to this of this microbe in the form of biofertilizer to the field. Now, this is I am going to discuss in the next one, that is how you are going to do the packaging in a carrier material. So, the carrier material is used as a vehicle to transfer those microorganisms which are acting as a biofertilizer from production unit to the agricultural field, right students. So, peat soil, lignite, vermiculite, charcoal, farmyard manure, soil mixture are actually used as a carrier material. So, do not forget these are certain names. Peat is quite famous, charcoal is again one such famous thing, carrier, lignite again. So, these are the carriers. You can see in this diagram also people are using these peat and other charcoal materials which can act as a carrier of these microorganisms. Okay, so what exactly the characteristics of an ideal carrier should be? It should be cheaper in cost, it should be locally available, having high organic matter content, no toxic chemical, water holding capacity must be more than 50%, easy to process. Okay, so you should remember that without a carrier material, your application of microbes is of no significance. Okay, so this slide is talking about preparation of those inoculant packets. So, neutralized and sterilized carrier material is actually spreaded on a clean, dry, sterile plastic sheet or metallic sheet. Bacterial culture which was drawn from the fermenter or bioreactor which I have just discussed is added to the sterilized carrier and mixed well by manual or mechanical mixer, and these are then packed in the polythene bag which is sealed with electric sealer. Okay. So, make sure there are certain things like inoculant should be carrier based, inoculant should be contamination free, it must contain 10 to the power 8 viable cells per gram within 15 days of manufacture. I mean, it is not like randomly you are providing the number of microbes, instead the count must be 10 to the power 8 viable cells per gram within 15 days from manufacture, and it must contain 10 to the power 7 viable cells per gram within 15 days before the expiry, right. इसका मतलब ये है that from the manufacturing till the expiry, the viability of the cells means the cells which are active, that is very much required at this rate, that is 10 to the power 8, right. So, ऐसा ना हो कि your carrier is without an active microbe, the microbe must be active, that is the meaning of viable, right. And packet should contain the name of the host plant, means जिसको वो provide करना है, किस plant को provide करना है, then name of the manufacturer, date of manufacture and expiry, and net quantity and storage. Students, I have discussed many a times that you must check date of manufacture and expiry every time either you are purchasing anything associated with that. So, this is seed treatment when you are applying mixing of seeds with the biofertilizer. Seedling treatment, when the seedlings are there, then you can see dressings and soil applications, right. So, you can use in either way like farmers are applying. You can see rice seedlings into this. This is the part where I am showing BGA, or in the circle part BGA or blue green algae, where blue green algae is going to help in the further growth of rice crop plant. Okay. So, dear students, I hope this is fine enough till today, and if you have any doubts, this is about how you will apply this azola, what you need actually, how much you are incubating, how much you are making the mat, harvesting it, drying it, and in this form you are applying it to the field. Even I will discuss some more in the next coming class, in the last class of the next week. Till that time, Thank you. Have a nice day.